Hello and welcome everyone in another episode of Researcher Celebrity. Today we have Mr. Vishal Sharma with us and a brief introduction of Mr. Vishal. He has done his bachelor's in life sciences and master's in microbiology both from MDS University Ajmer. Then he joined industry and worked in different industries, started with Lupin, Millipore, and now he has started his own, you know, VNI, a great entrepreneurial name also. And with that service oriented mind, I will say thank you so much for taking time out for this conversation.
It's a pleasure to have you. Thank you so much.
We always start our conversations that how and when you decided that you want to become a researcher.
So I'll flip the question why I decided not to be a researcher. And pardon me for a little bit of anecdote on that. Both my parents are PhD.
Mhm.
My father is a chemist. He did his MSc in Masters in Chemistry. Then he did his PhD in soil chemistry. He was a soil scientist. Few patents under his belt. If you know about some of those questions about now we are adding ethanol in our fuel, right? In petroleum products and all, and India has increased from certain percentage to new percentage. Questions are coming up, that could how will the old engines will take care of all that. Now where is that ethanol coming up? So all that ethanol research and some of those work which is being done was done by my father while he was and those are the patents what he had, and he was working very closely with the Ministry of Agriculture and a lot of these byproduct which are coming from sugarcane and sugar beet. So that's one side of it. And my mother, she is PhD in sociology, and she was not a typical lecturer of a humanities subject, but more of a researcher. And her topic of PhD was on child delinquencies and narcotics, and she has worked on some of those things, which became part of her PhD and became a little bit of controversy much before you have seen the movies like Urta Punjab and all those things. Uh, where early 90s, and she has published this entire research, which was about how narcotics are coming into Punjab and borders of Rajasthan, and how it is getting into getting access to the students, and how are they spreading, and what is all that. Uh, Part of it, I think India Today did some story in 1998 and quoted a lot of my mother's research in that, and her photo was put into that, and that became a big controversy. A lot of people who are from different government agencies descended at our home, and she was doing all the explanation. They were all asking, "Give us your sources and how did you do it?" And like a true researcher, she was true to her science and facts, and she kept it. Now, I grew up in this environment, and all my mother's friends were all researchers. My father's, friends were all researchers. Everybody is a doctorate, and I said, I don't want this life. Although I like science, I don't like this life. Everybody is. So our drawing room conversations would be about, whether a scientific thought should be applied to humanities and whether the concept of human psychology should be applied to chemistry. And imagine that kind of conversation, and your parents are not talking to each other for 2 days because of that. So I said, I don't want to get into all this. I cannot become a lab rat. I cannot become a researcher. I want to get away from all this. But I like science. I mean, I can tell you, I love science. I like science. But I wanted to use sciences for something else. There are so many other things, and that's why I was very clear that I will not pursue my PhD with lot of resistance at home, where both of them were heartbroken. My parents that I have such a promise and all those kind of a thing. Why can't I take forward the legacy which is there in the family? But I said no, I'm not going to do it. And then I think in my class I was in my senior batch or junior batch. There was a tradition. So you finish your MSc in MDS University Ajmer, and all of you go to CDRI for PhD, which is in Lucknow, right? So my seniors, there were around 4, 5 of them. My batch, I think 3, 4 of them went into that. My juniors and so on and so forth. And I was the only one who was actually searching for a job after MSc. I got one in Lupin Limited, Mandideep, which was a pharmaceutical manufacturing, and that is the first time I got to understand about application of science. And so To put across your first question, I can tell you why I'm not a researcher.
So you made this conversation already interesting. So let's say that with all this background, where parents, you have seen probably the suffering of researchers, okay,
Yes,
and then you were like,
very
okay,
early
I
in
do
my
not
life.
want. Yeah,
Yeah.
yeah. So you were like, I do not want to live this life. But
Yes.
that passion, commitment for science, which have driven you till here, and we see a promising future also. How will you say that if these researchers have not done all the fundamental work,
Yes.
or as you said, like the contribution of your mother, your father into these main hardcore, like different fields, obviously, but still that thing, how you have taken all the good part of that, that okay, you know how research works, and then what are the challenges which it comes after, as you said, either reports, publication, papers, or whatnot. How that
So,
worked for you?
so I think what I liked about is the scientific temperament, which is there in any research, because the question is why.
Yeah.
The entire science runs on the question of why. Why is this something happening? And we go about it, and that's something which got ingrained into me very early because of, I will call it as a privileged upbringing, because all I found out is not only my parents but all their friends were all those kind of people, right? But what I realized was that there is a lot of work which is required. I have seen some of the, when we talk about this ethanol, which has just come now, right, in India, where we are adding it into our petroleum products for vehicular movement. Now, imagine this is something which my father has worked upon in 90s, and the data was available for him, and he was doing a lot of those kind of work, not just on ethanol, but he was doing on. There was another. So if you look at it. With respect to sugar beet. Now, India, there was only one sugar factory in entire India which was making sugar out of sugar beet, which is in Sri Ganganagar in Rajasthan. And that is what my father was also support. Now, he was not working with sugar beet because it was a state government enterprise. But as a scientist, he was doing all the tech transfers and all those work from agriculture departments and ICAR and all those things, and he would know what is, because being a chemist, how much is the recovery and what are the byproducts. So some of those byproducts which were there, which was a problem, were pulp which comes out of sugar beet, and it is very rich in vitamin B12, but very 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 bitter. So he created gems and jellies out of it, because one of the reason was it is not the sugar beet was not becoming mainstream in India was because of the byproducts are so, I mean, are so much, and disposal of those things were costing more than the extraction of sugar. So he worked on all those kind of things. He developed distempers out of fly ash. He developed a process for creating gems and jellies out of very heavy, rich vitamin B12 sugar beet pulp after the sugar extraction. And of course, the ethanol, which is coming up because the yields were sometimes very high, can be utilized excess of ethanol, because at that time all ethanol will be going into the distillery for your we used to call them English wines, but they are basically blended rums, right? So you call whiskey, you call everything. Everything was rum. Everything is made of molasses in India in those days in 80s and 70s. So he would work on all those kind of a thing. How do you handle this excess, this kind of a thing? And he developed patents around it. He developed the process. But what I realized is that there is no connection between the research and a large scale application of that. And many of his work actually went, I mean, it took a lot of time to come into the mainstream, 30, 40 years, even after the overall, the process being developed and all those things. To get it into the large scale kind of a thing, there was no infrastructure which is available. There's no thinking which is available. And that is something which was intriguing me. What is industry thinking and what is scientist thinking? Somebody has to bridge both of it, because industry is for profit, and until unless something is profitable, there is no point in investing into something which is basic. But if there is nothing basic, how will you create your future profits? So that's one of the reason why being a scientist in me, and as I said, a part of it when I saw those struggles of commercializing any technology or a thought, it requires a lot of systematic work, and that itself is a scientific work, by the way.
Indeed.
Because most of the scientists think that you have discovered something, it can be done, let's solve it technically. But you have to solve it people wise, you have to solve it infrastructure wise, and you have to solve it technical wise. So there are a lot of those things, and that enticed me into getting this career. So,
Absolutely. So let's say that you're training at Lupin and then Millipore. When was this thought that you wanted to have your own entrepreneurial start?
okay. So I'll start with Lupin, and I'll come to it. It evolved. It did not happen in one day that I got a, the Moses came to me and gave me 10 commandments, and I started leaving Egypt to, to the Levant. I didn't do that. It is something which developed over a period of time. When I went to Lupin, I had no idea about pharmaceutical manufacturing. I had no idea about it, and it was sheer luck. We call it serendipity in scientific terms. It turned out to be the only USFD approved plant for sterile manufacturing in Asia, where I landed up for the interview. I didn't do very good in interview. I can tell you that. I mean, in fact, I was naive in asking some of those things. Some of it, I we the who was my the person who took my interview at that time and who later on became my boss and me. We laugh at those questions which happened at that time. The answers I gave, but somehow they decided to take me as a trainee. So this was 6 months training, and then you get confirmed. Then you get into probation and all those things. So when I went there, I was fascinated with the concept of utilizing microbiology because we have studied microbiology, and most of us who were in microbiology would go into fermentation or some kind of those kind of activity. Here it is completely opposite. You are basically finding out ways and means to kill microorganisms because you don't want any microorganisms in sterile manufacturing. So you are looking at so many of those things. The entire concept of clean room was something which was out of the world for me. It's like you're wearing a space suit with completely covered and getting inside the clean room and so on and so forth. So I was mesmerized with this entire thing, but I realized there are a lot of problems over there. One of the things which, and not immediately over a period of time when you learn, the biggest problem is how do you replicate? So you have here are people who are gowning and are doing some things, and every time when you are doing something, you are making sure that you are not disturbing the air. So you're very slow. It's something like very robotic movement which you're doing, and and suddenly if you start feeling a little bit of sweat inside, you have to get out of that place, replace your gown, come back. Now, if I'm working on it, how do I know that this speed is slow or this speed is slow? How will I get to know? Right now, you need to be constantly reminded about it, right? And a lot of training is required on that. Second thing is how when I feel that I'm sweating from inside, well, actually nothing is happening from outside. It has just started. The time delay, what I think versus somebody else thinks, could be very different.
True.
And those are the causes of contamination which happens in sterile manufacturing, and later on it becomes a real trouble for the company. And when you have to constantly review it and all those things, so how do you scale it up? How do you take it to everybody? Now there are technological answers for that, which is now the modern manufacturing is all about, where we have got isolators and other things. But before all that, how do you handle it? How do you train people? How do you work with people on those kind of a thing? And how do you make sure not by somebody will not do it purposefully, but omission error happens, right? You when you're working for 8 10 hours, you can forget something, right? So those kind of a thing, how do you work on it? And those are the whys which I started working on in my tenure in Lupin. Although not part of my regular job role, but it used to fascinate me. And that probably also propelled me faster in Lupin, by the way, as compared to a typical role profile point of view. Because they realize that these are those miscellaneous jobs which can be given to this person who will actually do something and come back with something on those things and try out something on those kind of a thing. So that freedom, I really appreciate the management there and those people who have given me. But that seeded a thought that there has to be something. Now, this is one lupin, one site, one location. How many lupins are there in the world? How many sites are there in the world? And during that time, filtration used to be a big challenge for us on installing the filters and everything. Now everything is automated. In those days, everything used to be manual. We have to do integrity testing of those filters and all those things. Now, that is the time Millipore used to supply all those things. They would come, they would train us. And I used to think, I need this job. I need to do this, what they are doing. And one day I asked, and that's how I met my future business partner, Ivy Lewis. She came to train us, and she's a pharmacist and MBA in HR. So I asked her, naive I was, I said, I want your job. And she laughed. And she said, okay, we have something coming up in validation, so why don't we interview? And the interview was scheduled with different people after some time, and that's how I landed up in Melipore. But when I got into Melipore, India was exploding. India was exploding. When I say India was exploding in 2002, 2003, with a lot of ANDA submissions to US, dossier submission to US for products to be supplied for US, Europe, and global. Products were coming from R&D to production, and those are the products which I was validating before it goes to production. And I realized there is a pattern of the same kind of problem which is happening whether you do company X or company Y. Now, these are all big names in Indian pharma, and you realize that all of them are doing the same. There is the same pattern of dealing with things. So the researchers, in our terms, we call them formulators, the formulation development scientist. And so there is analytical development scientist who will develop an analytical method, and there is a formulation development scientist who will develop a formulation, which you basically will either put it in the form of injection or tablet or whatever if you want to use it. So these formulation R&Ds would send me the formula and all those parameters what they are doing, and they have some kind of vision of the scale up batch. And this is what my lab has to do, is to see whether their vision of the scale up versus what they have done on the beak in a flask is actually going to marry in the production side of it. And I realized that they have probably not got that kind of exposure. Or now these are PhDs in pharmaceutical manufacturing. I mean, the pharmaceutical development and all those people, different types of people, pharmacists, chemists, and biotechnologists. These are all, and the products could vary from biological vaccines, oncology products, all kind of products used to come to me because everywhere filter is used. So I think I was lucky that I got to know viral clearances and so many things used to happen in that entire thing. But R&D scientist is basically throwing the product over the wall to the production, and production is catching and making sure of whatever it is. And we used to call them engineering batches in production where they will redo the R&D at last scale. So, for example, if you're talking about insulin, uh, so you have bacterial processes and you have yeast-based processes. In India, there are two, three companies. Won't get into all those details. These are all public domain information. Now, all of those places, these people would have worked on some, 500 liters or 200 liters of fermentation, and suddenly you are going to take it to 5 kl and 10 kl kind of a thing. Now, the kind of impeller movement with the kind of waves it is going to generate, or the vortexes it is going to generate, or the kind of heat generation will happen, will be very different. And what is happening in 500 liter versus 5 kl tank. And similarly, when you're looking at downstream processing and things coming onto filters, when you have done with the disk of 13 so in laboratory, you will have a very small disk on which you will filter something and see whether there is an absorption that is happening or not, versus it has become 10-inch device. Now, the kind of, which is around 7,000 centimeter square inside that, which is only, I mean, and what you have worked is only on 13.8 centimeter square. Now, suddenly, all this, the absorptions, the loss of products, everything will become very different than what you're seeing in your R&D spaces. And there was no connection between these two groups. So, all R&Ds of all the parameters, specifications, batch manufacturing formulas will undergo invariable change at the production level than what you get into R&D. So initially, when I got into it, when we started this lab, we would take whatever R&D is given as Bible and would do the validation. And when this validation goes to the production, the validation fails because their parameters are not even coming anywhere closer to that. And that has taught me that this is a bridge which has to be looked at, worked on it. But around 2006 and 2008, what happened is most of these products which were in R&D and where we have done the submissions to US, Indian companies have done submissions to US and Europe, they started getting into manufacturing. Now, because the approval takes 2 to 3 years of a product. So after you submit your entire dossier, questions come, review happens, and all those things, which means you have submitted all your science, all your data, everything is done. Now the review comes. I mean, it takes, depending on whatever it is, between 9 months to 3 years. But 2006, all these race which was happening between Indian generic companies in their R&Ds, now the race is in the production, where everybody wants to sell the commercial batch to US because whosoever can reach there first will make the maximum profit. And everybody is same because everybody is working on the similar products or same products portfolio, whatever it is, the generic products. So and that is a time if you see in Indian pharma, there was a lot of first time there are a lot of regulatory action started happening. So you have US FDA giving warning letters to Ranbaxy used to be the number one company in India for 15, 20 years and way ahead of everybody else. And today we don't have Ranbaxy. The name itself is gone. They had got into warning letters, then they got into consent decree, and similarly, warning letters and data issues and all those things started cropping up in different Indian companies. Now, these data issues, although if you read and everything, it looks like fraud, it is not. It is this problem where the scientist or researcher and the applicator or those who are applying it, they are not talking to each other. And once you have submitted, the system of regulatory submission is once you have submitted a specification, now if you have to change it, it takes another 3 years. Now here is where the MBA comes in. We don't care. Do what you have to do. You know, the management, the MBAs, they come up. We don't care. We have spent 6 years, 7 years on this product. Now's the time to harvest it. And you are saying we have to resubmit, which means we have to wait for 3 years. Market is gone. So this catch is what started us to look at that science is not just filtration, but it is in all kind of facets of pharmaceutical manufacturing, and that's how Vi'eNnI started in 2010. Saying is easy. I kept my resignation letter for 1 year in my pocket, attended Melipor's long range planning because I was one of the fast track managers in global Melipor, what they have looked at, and I was supposed to take over certain other functions as well. So scientific functions, of course. So I actually have attended a long range planning in Kuala Lumpur, which was done for all over the world, and made a plan in 2008 for 2035. Till 2035, how will we grow? What kind of technologies we'll need? With my resignation letter in my pocket, and saying that I need to start. But 2010, finally, we started it, and 15 years is a roller coaster ride.
So this is the point where we always talk in our conversations that, as you pointed out, that whatever works in lab, when it goes to production, the things looks like it's all over a new story.
Yes.
It feels like, as you mentioned, that this gap. So how does VNI fills that gap?
Okay, so what we do is we are very uniquely positioned as an Indian company. Mostly, this is dominated by multinationals in this domain, but we have our operations not only in India but outside India, and Europe, and US as well on these aspects where we do. So what we do is we basically look at something in pharma terms. We call it a cycle development or the process development. So
Hmm.
you have, and by the way, the companies do have their own teams for that. These are called as MSNT, Manufacturing Science and Technology, or MSTG, Manufacturing Science and Technology Group. So they will have all kind of engineers, pharmacists, researchers, scientists, all kind of people, microbiologists, all kind of people will be there who are actually responsible to take this product to the shop floor, and they do a fairly good job, no doubt about it, because this has evolved in last uh, 20 years, but in India it has evolved in last 8, 10 years, and now there are several groups in Indian companies itself, their full-time job, and they are drawn from both R&D and production, and this is where it is. They are being put together, and they are doing very good job on that, this entire thing. But what happens is there are certain niche areas where you need a lot of understanding. I'll give you an example. Now, there's one thing which we have done it for one of our customers, and they have patented this globally. This product patent which they have got it. So let's talk about sterilization. We all know autoclaving in microbiology. We do a lot of autoclaving, and in biotechnology and all those. I mean, except in chemistry, any life sciences you have to do, sometimes you have to deal with an autoclave, right? So one of the ways of sterilization is autoclaving or moist heat sterilization, and in pharmaceutical manufacturing also, especially when you're looking at ophthalmics, eyes, eye drops, many of them are steam sterilized. So you fill it in those a three-piece bottle or a form fill seal or a blow fill seal, what you see as cartridges, and they are subjected to autoclave. Whereas many of these biological products, you cannot autoclave them, so they go through aseptic processing, filtration, and all those things. Now, this is an ophthalmic manufacturer, a generic manufacturer, ophthalmic manufacturer in India. Now, their product was something which cannot even withstand a temperature of. Sorry, so they were actually challenging a patent. So when they were challenging a patent, what they were trying to go around it is the API which is coming in the form of powder, which you will mix it in the liquid and then you will fill it in the bottles. I'm not saying that it is going to happen by pouring, but there
Hmm.
are big machines and all those things and big vessels around it. But this powder which is coming is supplied by only one, we call it as API, active pharmaceutical ingredient, which is a drug which is to be used. Excuse me. Now, this is coming, there is only one supplier. And this supplier holds the patent on it. And this is the supplier who is also having the formulation of it. So this supplier is not giving it to you. So they developed that API manufacturing backward integration, and they had in their API manufacturing, they come up with their own API, uh, generic API of that. Now this generic API, now the original manufacturer can give you sterile API, which means he is sterilizing it at their own place. Whereas this plant, where this entire thing was retrofitted, you cannot put a sterilization process. So this powder is coming to the formulation plant, which is another plant, where you are going to, fill it and all those things, mix it and fill it. Has to sterilize it, this powder. Now, this powder, I'm using the word powder, basically
Hmm.
substituted with the API, cannot withstand any temperature above 120 degrees centigrade. Now,
Hmm.
typical requirement is 160 degrees centigrade for 2 hours. Otherwise, regulators have questions. So if you
Hmm.
do not sterilize it for say, 160 degrees centigrade for 2 hours, dry sterilization, the which is under oven basically, we call it as dry sterilization. So that if it is not done, it is not acceptable. And here the powder has a melting point at 122 or 123. Now what do you do? So we developed a cycle at 111 degrees centigrade for them, and we worked on this entire thing where we proved the sterility, which is where microbiology comes up, the
Hmm.
effective microbiological removal of that cycle on that, because we know heat kills. Heat kills at 100 degrees centigrade also, maybe not so fast as 116, but it kills. Now, how do you work on this entire thing, and how do you look at reducing and eliminating their bioburden and achieving a sterility assurance level of 10 per minus 6? We developed this cycle.
Hmm.
This cycle went, first of all, convincing them that an Indian company and an Indian consultant can do that for
Hmm.
you, and you can actually challenge something in US as a patent kind of a thing. Itself was a big deal. But after that, once they agreed on it, we worked on it and we submitted this entire thing. I will say regulators were very, I think, generous because they liked this new technology which is coming up, and they also gave us their input beforehand. Before your submission, you can always ask for meetings. That this is something which we are doing. What do you think about it? They will not say we say yes or no to it, but they will tell you what all they would want to see in it. So based
Hmm.
on incorporating their requirements, we basically developed this. 2013, I think this product went to market, which means to the filing, and 2016 the commercial started, which means they got approval. And this particular part, what we did for them, because we did it for them,
Hmm.
they filed a patent and they got the patent on that. Now they hold the patent and they are now putting it across to other companies. This is what can be done. So this is how we work. This is one of the examples. So we take care
Hmm.
of the niche problem which is there in the pharmaceutical manufacturing. Take that problem or troubleshooting activities, and we get ourselves involved in that.
Absolutely. And I think this is what, when we say in these conversations, researcher, this is what it is. Customizing the solution as per the problem.
Yes,
Whatever is the problem should be challenged in that way, and then the solution should cater that, instead of, as you mentioned, instead of going and beating any other bush and then do that, that
right,
as you mentioned about the scientific temperament is what
right.
it makes a person a researcher. It has nothing to do with the designation, the degrees, or anything else. One
I
of the,
completely agree.
as you mentioned, when I was doing my master's dissertation, one of the drivers gave me a solution for something, and I was like, man, the only thing he was able to think like that because he was spending so much time with scientists, and the way they look at things, the way they come up with the solutions. And obviously, like from your childhood, that is what you got in genes, in training, in home. So you are helping VNI. So would you like to say something about VNI in general?
So V and I basically came from the term we and I, we as people and I as an individual. And what we believe is that if you follow a process to give input to the people, they will make the processes much more robust. So for any kind of an activity, there is one side which is process, and in process I'm putting facility, equipment, and technical know-how, methods, everything in one side of it, and the other side is people. Unless these two things marry within each other, you will not have a very robust process which is consistent and which is sustainable. So our entire focus from last 15 years of existence is on this direction, and that has also pushed us to participate in another. I was telling you before this interview, into a not-for-profit scientific body, which is called as Parenteral Drug Association. This was established in 1946, currently headquartered in Bethesda, with 26 global chapters. India being a chapter from 2013, and I'm one of those founding members for India chapter. Now, this scientific body is exactly doing the same thing, and we find a lot of synergies in that. Basically, what this period does is it publishes technical reports. Now, these technical reports are around a unit operation. We use the word unit operation in pharma, which means sterilization, autoclave. So you'll find a technical report on autoclave, which starts from how do you develop a cycle, what kind of troubles you will find it, and how do you validate it. So the entire thing is around 120, 30 pages, which is practical cases which are being put across over there. By the way, to put it across, what I was talking about earlier about this dry sterilization process at a very low temperature of 111 degrees centigrade. Uh, I am also part of a task force which is currently revising a PDF technical report, which is called Technical Report 3, which is on depyrogenation and dry sterilization. So the
Mhm.
work what I did is now getting published in that, and which will become kind of a quasi I will call it as a regulatory document because by design, all PD technical reports are reviewed by regulators, fixed regulators by design, if they are not part of the task force. Many times you will find them as a task force as authors who are working on it. If they are not, then they will peer review it. So, looking at whatever we have done is going to be part of that tech report 3, which is coming up in that, and it's a fulfillment of it of sorts. So, answering your question, research can happen anywhere, and
Mhm.
the ideas can come from anywhere and
Mhm.
from different kind of people. Now, why I'm talking about this tech report 3 on dry installation, and I'm talking about my work in 2013. One of the questions which has come up from this one of the person who is based out of Europe, because it has global, people, and you will find researchers, applicators, and people like me, miscellaneous who don't fit anywhere. So, so all kind of people are there in that. One of the questions which was asked about is if you are talking about this in this particular thing, why can't the same thing be done on endotoxins as well? So I'm talking about microorganisms, and this person started saying, can't we do something on? So you're talking about bioload. Let's talk about pyrogen load. So can we work on that? And this is an idea which has come up, and now we are researching on it whether there is already available research available, and can we kind of do a scale up on that, and can we see if it works in this way, and with different people across the world running the same experiment on a commercial scale and see if it works. So then we can actually change the established norms from last 70 years. No, not 70 years, 50 years. 50 years, there's only one test which is called as LAL test, Limulus amebocyte lysate test for endotoxin, which is a specification in USP, IP, BP, EP, JP, any pharmacopoeia in the world. Can we change this concept of it? So let's see what comes out.
Nice, nice. This sounds like, you know, been utilizing all the information which is there and building you using them as building block and making new buildings now. That is what VNI essentially is doing and catering.
Right. Right.
So one more question which comes is like whenever we talk about these differences, as you mentioned, the one who's developing it, the one who's using the applications and all of that, what is your uh, take or experience when you have worked for almost more than 3 decades here, that what is the biggest flaw whenever a development scientist is doing either it is a process or a product when they're developing, and whenever it goes to the market, what is the biggest thing which they miss or do not understand well?
Knowledge transfer. I think this is the um, uh, biggest challenge which is holding us back. Otherwise, a lot of things, a lot of problems which we are facing, we would have solved it 20 years back. And probably somebody has solved it already 20 years back. And I will talk about from my industry, but this could be the same thing with the different industries and different scientific temperament. And I'll come to it. This simple thing. COVID has, and I'll make it a very simple example from COVID because everybody can relate to COVID, whether you are from pharma or not. So during COVID, it taught us that we all have to wash our hands, right?
Mm-hmm.
And there was social distancing has to be maintained, and all those kind of things have been told to us. And I'm taking, pardon me, I'm taking a little bit of spiritual but very scientific journey back. Now, look at it. I come from India, and I will use the term not just India but South Asia, and irrespective of religion, whether it is Christianity in South Asia or whether it is Hinduism, Jainism, Buddhism, Sikhism, or Islam in South Asia. First thing which is being done is before entering into any place of worship, we remove our shoes.
Mm-hmm.
Whether it is Muslims, whether it is Sikhs, whether it is Hindus, we remove our shoes. Second,
Mm-hmm.
we wash our hands. And
Mm-hmm.
if you see even the wazoo, what Muslim do, it is not like just this. They have to actually scratch everything,
Mm-hmm.
and they do scratching in all the holes. Same with Hindus. We have to do all with our hands completely and with ash. I mean, we used to call it as rak,
Mm-hmm.
which is ash. You have to do all that particular thing. Same with your feet. Why would they do that? And we actually forgot because those people, it became a religious practice. And I'll use this religion later on with the way of life, what happens in scientific terms, and later on in our modern systems. The reason for using the ash was because it is activated charcoal. And activated
Mm-hmm.
charcoal does the same thing of binding with impurities and taking them away. You use it today for removing color impurities and all the pyrogens and all different kind of a thing in industrial scale processes. But for example, making your sugar white, activated charcoal is used along with other things, sulfur, and now sulfur cannot be used, so activated charcoal is used. So to remove all the impurities is where we use activated charcoal, and ash actually was doing the same job for us to remove all the microorganisms from our hands. And
Mm-hmm.
everybody knows whether you are going into the temple or whether you are going into the mosque or you are going into the gurudwara, any of those place of worship in India, we are in subtropical and tropical environment. A lot of microorganisms are there on our body, and we'll all congregate in a very small space. So you need to be taking care of your personal hygiene.
Mm-hmm.
And that's what, because you will be shaking hands, you will be doing namaste, you will be actually doing whatever it is, you will be touching each other, right? So before that, your hands need to be clean.
Mm-hmm. Nice.
And that's being done irrespective of religion. And COVID taught us that we have to do it. Now, let me come to it. Nobody told from that time to the passing generations the knowledge why we are doing it is lost. We started
Yes.
doing
That.
it as an act of mechanically, we started doing this, and we made it as a lot of casteism and all those kind of a thing, and everything moved into it. That's a different story altogether. I'm not getting into that. But why we do it is nowhere informed to us, and we all forgot about it over a period of time till COVID told us that you need to maintain social distancing and all those things and wash your hands and everything. Because poor guys did not have antibiotics in their time, and they knew this is the only way to control the disease. Now, coming back to the from R&D bench to the commercial scale, what happens? We send the entire protocols, we send the entire information about how to do it. So you have steps. This is what you do: mix this, mix that, rotate it for 15 minutes, then do this, then do this, then do this. But nobody puts across why we are doing it. And that's where the explicit knowledge, and in knowledge transfer there are two things: implicit knowledge and explicit knowledge. The explicit knowledge comes to the the next the people who are downstream, but implicit knowledge never transfers. And therefore what happens is at some point of time you reinvent the wheel, you get to understand everything, and then you redo it, then you again forget, again redo this, and that's the cycle. So the biggest
Yeah.
challenge is to transfer the implicit and explicit knowledge, which will make sense to people when they're getting into trouble.
This is so good. And I will say to all the audience that if you are planning for going to market, or whatever you are developing, these are the things which I think now you should take note of. And Mr. Vishal obviously is working great with VNI. We'll say thank you so much, Mr. Vishal, for sharing this journey. And I'm sure now whosoever is going to listen to this conversation will have two or three more steps in their head to take care of whenever they are developing something with this mind that they want to serve the society.
Thank you so much for having me, and thank you so much for allowing me to share my some of those. I will call it as wacky thoughts. And as I told you, somebody who has run away from research, if I could share something with people who are researching it, if I could be of any help. But thank you so much. Thank you so much for having me.
It was a pleasure having you, and to all the audience, will say stay tuned for another episode of Researchers Celebrity.
Thank you. Thank you so much.